Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Good morning everyone, we are from Butu. I'm Licea Deviska.
Tashila Tania, Diva Arifah.
Today we will explain what proteins exist.
We can think of this in terms of two steps, transcription and translation. In a nutshell, transcription is the process of taking a single gene of DNA and copying it into a structure called mRNA. Then translation is the process of taking this mRNA strand and using it to produce a protein. Before we go through the details though, we need to look at why we actually need these two steps. Inside almost every cell is a nucleus that contains all the genetic material of that cell in the form of DNA. When we say DNA is so essential to life and controls what cells do, is because it contains thousands of genes, which are each small sections of the DNA that have a specific sequence of bases, and so are able to code for a specific sequence of amino acids, which when combined will form a meticular protein. In order to actually make a protein, the specific sequence of bases has to be read by one of these structures, called ribosomes, which importantly are outside of the nucleus.
So as I was saying, ever since I switched to I felt empowered.
But because the DNA is so big, it can't leave the nucleus itself. And so if we want to use a gene to make a protein, we're going to have to make a copy of that gene first. And because we're only copying a single gene, rather than the entire DNA strand, the copy will be small enough to leave the nucleus, and so it can make its way to the ribosome. Now, this copy we're talking about is mRNA, which stands for messenger RNA, and is just a copy of a single gene. The structure of mRNA is mostly similar to DNA, but it does have a few important differences that you need to know about. For one, it's much shorter than DNA, because it's only a single gene long. It's also only a single strand, rather than a double strand like DNA. And finally, instead of containing the base thymine, it contains uracil. Now that we've got these basics covered,
We must know that protein synthesis is to making a protein. There are two terms, translation and transcription. Transcription is the process taking a single gene of DNA to be a mRNA. This is the DNA. It happened in nucleus. And translation is the process taking mRNA in ribosome, the process taking mRNA to produce protein or amino. For further material will be explained by my team.
Okay, now let's explain about transcription. So in transcription there are three steps. The first one is initiation. For initiation, the first one is the DNA strand will be from the helix. Jadi kayak tahu tuh tergolong kan, terus dia jadi kayak kebuka, terus dia there is RNA polymerase, an enzyme that will bind to the DNA to the gene that you want to copy. The name is promoter region. So this is like the enzyme that clean RNA polymerase. Okay, the video is showing you the second step, which is the elongation step. This one is the initiation. And for the elongation, so this is the DNA, right? This is separated by the RNA polymerase, and then there are two types of strand. The first strand is the one that is non-template. That's one. Wait, non-template. This one will be not used for the reading the code. Jadi namanya itu non-template code strand ataupun coding ataupun sense. Sedangkan ini sebaliknya, itu tuh namanya template strand ataupun anti-sense. Nah, jadi kan kalau misal di non-template strand itu dia start dari 5N ke 3N, sedangkan yang ini tuh 3N ke 5N. Nah, kalau misal ada RNA-nya tuh, dia tuh bakal start dari 5N ke 3N juga. Jadi kayak sebenarnya ini sama ini itu komplementari, jadi kayak hasil kodenya itu sama, tapi kecuali di beberapa part misal kayak kalau misal di sini ada A kan seharusnya di sini P, cuman karena di sini nggak ada RNA itu nggak punya dan doesn't have timing, dan they use Brazil gitu. Jadi kayak sama di sini ini sama ini itu complementary, tapi bedanya cuma di bagian timing dan replikasinya. Jadi perlu kalian tahu kalau misal mRNA itu dia mRNA start from 5N to 3N.
Just ahead of the RNA polymerase and closing just behind it, so that only a small section of the DNA is ever exposed. Once it's moved along the entire gene and finished making the mRNA strand, the RNA polymerase detaches from the DNA, and the DNA strands enclosed by that. This means we're left with an mRNA that's then free to leave the nucleus and head off to the virus. One last thing to mention though is that this strand of DNA, which the RNA polymerase moved along, is called the template strand. So it's the template strand which is used to make the mRNA. So going back to our whole cell project, we've now.
And then the last step, it is termination. In this step, the RNA yang udah kebentuk itu bakal jadi RNA polymerase nya itu bakal separate gitu. Separate, terus DNA-nya tadi yang pertama separate itu bakal gabung lagi. Nah, kan ada reaksinya. Misal ini mRNA. Tunggu, ini namanya tuh pre-mRNA. Nah, dia tuh ada beberapa section. Yang section yang nggak kebaca code-nya itu namanya itu introns. Nah, ini introns. Sedangkan yang kebaca code-nya itu namanya exons. Nah, jadi this one introns being removed and the exons is remain. Nah, jadi kan kayak orang ini udah terbentuk, jadi kayak gini kan. Ini udah yang kebaca, yang kebaca semua. Sama di sini dia di cap dengan di 5N itu di recap dengan sesuatu recap sama yang di N3 khusus N3 itu dengan namanya nah ini namanya polyetil. Polyetil itu dia dari enzim polymerase. Nah setelah itu guna cap ini tuh dia protect strandnya itu dari being degraded by certain enzyme, jadi biar nggak kebaca ataupun digabung sama enzim-enzim lain gitu. Nah setelah itu polyetil juga berfungsi untuk membawa mRNA ke luar dari nukleus untuk menuju ribosome ke cytoplasmus. I have some practice for how to read the mRNA. So ini kan misalnya ini 3, ini DNA-nya template strand, sorry. I want you help to complete this RNA code. So the first one is then the second one is if it is in the non-template strand, it will be T1. Because it is RNA, it will be Russell. U and and then the next step, which is translation, will be explained by Diva.
Now we've got an mRNA copy of the gene, here inside the nucleus, which is free to leave the nucleus and make its way to the ribosome, where it can undergo translation to produce a protein. To help understand this part, it's important to remember that for both DNA and mRNA, each group of 3 bases, which is called a triplet, or codon, codes for a specific amino acid. To make proteins, our cells use 20 different amino acids, and each one has a different 3-base codon. For example, this triplet, AGU, codes for the amino acid serine, whilst CCA here codes for proline. You don't have to remember these examples, we're just using them to illustrate the point. So let's now zoom into a single ribosome and go through the process of translation. To start the process off, our mRNA strand and the ribosome both bind together. And we're now ready for the ribosome to start building the protein by adding one amino acid at a time. The amino acids themselves are brought to the ribosome by molecules called tRNA, which stands for transfer RNA. tRNA molecules have the amino acid at the top and an anticodon at the bottom. The anticodon is this sequence of 3 bases, which are complementary to these 3 bases on the mRNA. And it's these 3 bases.
Okay, so first then we have translation. So what is translation? So translation is the way of the sequence of mRNA. So translation is where mRNA is a sequence of mRNA becoming into protein through amino acids. Protein through amino acids. First, there is the RNA has a sequence. You can just call this as a strain of RNA. And we'll bind first with the ribosome. Just three of this is ribosome. And then we have here, I will write just one more. Here, this is an tRNA with a sequence of, this is a codon. This is the a codon of an tRNA and has their own amino acids. And each tRNA has different codon and also a different amino acid, and each, etc., etc. This is an amino acid, any kind of amino acid depends, and a codon. A codon, I would just say maybe like an A, or U, something like that. And it has three, which makes it into a codon. This three is called anticodon instead, which in our mRNA is instead called codon, which is a sequence of three base, yeah, which is a code for an amino acid.
So because each type of tRNA molecule is specific to a particular triplet on the mRNA, it can ensure that it always brings down the correct amino acid. For example, because the first triplet of our mRNA sequence here is a GU, it will attract this tRNA molecule with the anticodon UCA, because UCA is complementary to AGU. And the tRNA brings with it the amino acid serine, because AGU is the code for serine. Meanwhile, this second triplet, CCA, will attract this tRNA molecule with the complementary anticodon of GGU, and that will be carrying the amino acid proline, because CCA codes for proline.
Okay, so to this point, the tRNA will bind with the RNA as they come into the ribosome. I will make this with the three codon. Oh, I have it down wrong. Three codon with their own amino acid. For example, in the video, this amino acid is called serine with a code of UCA. In the mRNA, it is AGU. Okay, this makes it will be the UCA and the AGU will each other become complementary. Now, another tRNA will also come into the ribosome per their complementary codon and anticodon. For example, here there's proline. Can you look at it? There's another one here, which is GGU with a complementary from mRNA, CCA. This is a tRNA for the amino acid of proline.
Because CCA codes for proline. Now, the whole point of this seemingly complex process is that the tRNAs have now brought the correct amino acids down to the ribosome in the correct order. So the ribosome is able to join them together and start building up a chain of amino acids. Once it's joined these first two amino acids together, the ribosome moves along the mRNA slightly. And so another tRNA molecule will come down and binds to its complementary codon on the mRNA, bringing with it the next amino acid. This allows the first tRNA molecule to detach and repeat the process. But importantly, it leaves the amino acid behind. This same process then repeats all the way along the chain. until the ribosome reaches the very end and has joined together a complete chain of amino acids.
Okay, so we first have the first tRNA and another tRNA. This will go down again and again throughout the RNA, while also the ribosome here, the ribosome will also move according to the RNA that's been sequenced with the mRNA. But thus, this tRNA will also move along the process as the ribosome would move across the RNA, thus only leaving the amino acids along. And here would form a chain called chain of amino acids, or it's also called polypeptide, which is a chain of amino acids.
At this point, the amino acid chain will detach from the ribosome, and then finally the chain can fold up on itself to form a protein.
And then this is the final step, which then all of the amino acids, which along with polypeptide, would form into sort of a ball kind. They would formed up into this type of folding ball, which we call now a protein. So this how we get our protein in the photosynthesis process. And then now we have some summarization from our dear Tashila. Thank you.
So I will review our recall in a short way, so maybe you will deserve to remember. So the first one is in transcription, that is imitation. The imitation step is when the polymerase, RNA polymerase will join with the DNA. So this is the imitation step. The second step is translation. The translation is when the RNA polymerase separate the DNA, so it can read the DNA strand. Right. And then the third step is termination. That is when the RNA polymerase is separate and become to mRNA is jadi sudah terbentuk. Jadi baru di termination. And the second method is translation. That is, the first one is when the RNA will break amino acid in the ribosome, and the mRNA will be here too. And then the ribosome will read the mRNA triplet by triplet, like we are doing yesterday when we are read the triplet. That is the job of ribosome. And then at the end, after it finishes, it will be amino chains and it will be to protein.
Okay, that's it for our presentation. Is there any questions? I'm feeling like there's something in Lisa's eyes. Okay. Oh, okay. First Nabila, because she's been.
And you explained in the translation.
Oh, okay.
I think you explained that like that lah. But I confused that there is start or continue with long until what? Is there a start or the codon? Is there the start or the stop?
So it depends with it really depends with the triplet. The start of the starting triplet will be IUG, and then the end. IUG is the starting triplet. And then the ending triplet will be, there's 3 triplets, which is U, U, A, U, A, G, U, A, G, and U, G, A. This is in mRNA stand. In RNA stand. This is the stopping triplet. And this is the starting triplet.
Thank you.
Lisa?
So I'm kind of confused about the evolution of evolution. Like if we're like from 5 to G or like from G to 5. So before we learned like the introduction, some introduction about the nucleic acid, and then in some strand, something starts from 5N to 3N or the kebalikannya atau dari 3N to 5N. I think this already kayak udah pasti gitu. The one that yang kebaca, maksudnya yang template strand itu udah pasti N itu di start dari 3 dan satu lagi itu diakhiri dengan 5. Jadi antar mRNA-nya itu bakal 5N to 3N. Itu emang kayak pasangan itu. Jadi kalau misal di yang satunya 3N ke 5N, pasangan satunya itu dari 5N ke 3N gitu.
Anything else? Okay, Miss maybe. Oh, that's right.
So in the video, I saw that like in the transcription stage, it says that like the gene that we want to copy. Does that mean
The gene that we want to copy.
Yeah.
Copy in that.
Yeah, does it mean that we can choose a gene that we not we, but like in the process that it can choose the gene that doesn't want to be copied?
Oh, in the video, when it says the gene that we want to copy, I think it has like an example because it's really long, right? Of course, it will start from the start of the mRNA, but it really depends on how the triplets are started, right? So yeah, it will start really on the actual reading that will be able to be read will depends on the triplet, if it's an AUG, and where it's stopped, which is these 3 triplets. Yeah.
Thank you.
And us is Liz, I think you had a question.
Okay, good. Finish? Okay, let me ask, please make a simple conclusion of a whole process of the protein synthesis by your group. I mean, wordy. The explanation is wordy, so I need the conclusion. What is the simple way, how you make a simple way conclusion from your
So in, do you mean like in a more shorter way? Should we say about transcription versus transmission? Okay. So in the process of protein synthesis, we have there's two process, which is transcription and translation. Transcription is
stitching DNA double strand into single strand, which is called as mRNA, and then for the translation, making protein by reading the mRNA codes to make an amino acid and then protein.
yeah.
And then how about the enzyme? Is there any enzyme that involved?
In transcription, there are 3 enzymes that involved. The first one is RNA polymerase, and then that will binds to DNA, and then to separate the DNA, and then the second one is polymerase, that is for the tail of the 3 ends, cap the mRNA to protect from the others, to protect other enzyme to mengganggu mRNA tersebut. And then the third one is
Please, or something.
That's one is for the to to split the
Introns and exons.
exon and introns, and then make the exons to bind.
And by each other.
How about the translation? Is there any enzyme that involved in that process? You can explain by your own book. I mean, if there is no or you didn't find any enzyme that written in the file, it's okay.
From the file itself, does it explain about the enzyme that is involved in the translation?
But I believe like for example with your video history, I believe there is an enzyme. For example, we have a to transfer mRNA, or yeah. But I haven't actually checked your videos, so I could not see any enzyme which in translation in this video.
And the last one I want to ask to pick up. Maybe you can make a simple conclusion. The protein synthesized starting from the process, the steps, and the place where it happens.
Transcription is the process of taking a single gate of the DNA to be mRNA and happen in nucleus. Then, if translation, the process taking mRNA to produce protein or amino acid strands and happens in ribosome.
Where do we find the ribosome in the cell? Reticulum ribosome. I mean, even reticulum ribosome. Where do we find the reticulum ribosome?
Outside of
What is the name of the outside of nucleus? Yes, cytoplasm. So you will find the ribosome in the cytoplasm of the cell. Okay, thank you. You can close.
Okay, so that's our presentation. We apologize if it's too wordy and too complicated with very poor drawing. But yeah, we hope that you understand our presentation for today. And thank you for your attention today. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.